Hi, Mrs. Young here, and welcome to my podcast. Today, we're going to be talking about the amazing cell membrane and the reason it's so cool. The cell membrane really is a very important part of cells. But first, a little video about how the cell membrane was discovered.
Every cell in your body is separated from those around it by its outermost layer, its membrane. A cell membrane must be both sturdy and flexible. Imagine a membrane made of metal, great at keeping the cell's guts inside, but horrible at letting materials flow in and out. But a membrane made of fishnet stocking would go too far in the opposite direction, leaky but easily torn. So the ideal membrane falls somewhere in the middle. Over the past few centuries, we've learned a lot about the way membranes work. The tale starts in the late 1800s, when, according to legend, a German woman named Agnes Pockels was doing dishes. Her observation that not all detergents dissolve grease in the same way piqued her curiosity, so she made careful measurements of the size of soapy films that formed on the surface of a metal tray filled with water. Later, in the 1920s, GE scientists Irving Langmuir and Kathleen Blodgett re-examined the problem with a more elaborate contraption and found that those tiny slicks were in fact a single layer of oil molecules. Each oil molecule has one side that loves water and floats on the surface, and one side that loathes water and protrudes into the air. So what does it have to do with cell membranes? Well, at the turn of the 20th century, chemists Charles Overton and Hans Meyer demonstrated that the cell membrane is composed of substances that, like oil, have a water-loving part and a water-loathing part. We now call these substances lipids. In 1925, two scientists, Evert Gorder and Francois Grendel, pushed our understanding further. They designed an experiment meant to test whether cell membranes are made of only one layer of lipids, a monolayer, or two layers stacked on top of one another, called a bilayer. Gorder and Grendel drew blood from a dog, a sheep, a rabbit, a goat, a guinea pig, and human volunteers. From each of these samples, they extracted all the lipids from all the red blood cells and placed a few drops of this extract on a tray of water. True to form, the lipids, like oil, spread out into a monolayer, whose size Gorder and Grendel could measure. If they compared the surface area of that monolayer to the surface area of the intact red blood cells, they'd be able to tell whether the red blood cell membrane is one or two layers thick. To understand the design of their experiment, imagine looking down at a sandwich. If you measure the surface area of what you see, you'll get the dimensions of a single slice of bread, even though there are two slices, one stacked perfectly atop the other. But if you open the sandwich and place the two slices side by side, you get twice the surface area. The Gorder and Grendel experiment is basically the same idea. The open sandwich is the monolayer formed by the extracted cellular lipids spreading out into a sheet. The closed sandwich is the intact red blood cell membrane. Lo and behold, they observed a 2-to-1 ratio, proving beyond a shadow of a doubt that a cell membrane is a bilayer, which when unstacked yields a monolayer twice its size. So almost 30 years before the double helix structure of DNA was elucidated, a single experiment involving fancy versions of household materials enabled deep insight into the basic architecture of the cell.
So let's talk about the structure of the cell membrane. It all starts with a lot of phospholipids. So as you can see, and like the video talked about, a phospholipid has a hydrophilic head, meaning it loves water, it's attracted to water, it wants to be next to water. And then a hydrophobic tail, meaning it repels water, it hates water, it doesn't want to be anywhere near it. When you get a bunch of those phospholipids together, that's when you get your phospholipid bilayer, as they call it. You have one layer of phospholipids on the top and one on the bottom. As you notice, you can see how they're lined up. You have water on each side, water on the outside of the cell and water on the inside of the cell. So the hydrophilic heads, they're pointing outwards because they love the water. They want to be next to it. Then on the inside, you have your hydrophobic tails. They're as far away from the water as they can get. So sometimes you have to have something, well, not sometimes, all the time. You need something to help keep those phospholipids together. So if it gets really cold, or the cell membrane gets really cold, or it gets too hot, those phospholipids want to move. We have cholesterol here. That keeps the phospholipids the perfect distance from each other all the time. It's flexible and allows them to move a little bit, but not too much. In addition to cholesterol, there are more amazing wonders residing within the cell membrane. Proteins. Different types of proteins call the cell membrane home. Proteins help to move the larger materials in and out of the cell. So cell membranes are selectively permeable. What that means is just a fancy word for some materials can pass through while others cannot. So as you can see here, here's the cell membrane, and these little particles like oxygen or carbon dioxide or water can easily move through or move out of the cell. But these bigger molecules, like say a glucose molecule, or these look like possibly red blood cells, they cannot go in and out as easily. So I hope you enjoyed the podcast. Thank you for listening and have a great day.